In the previous video, we discussed about the Cas9 structure and the functions of the Cas9 enzyme. Now we will see the single guide RNA, its formation and the structure of the single guide RNA. So we know that this CRISPR RNA and the tracer RNA, when they get bind together, they form the single guide RNA, and we denote it as the gRNA or the sgRNA. Now these two RNAs, they are connected together with the help of a loop that is called as the connecting loop, and that is why these two RNAs, when they get bound together by the help of the connecting loop, and they form this structure that is called as the single guide RNA. And we know that this guide RNA loading, it enables Cas9 to form a DNA recognition competent conformation for target search. Now there is a part of the Cas9 that is called as the dead Cas9, or we denote it as the dCas9, and it can bound to the target site, but it is unable to cleave the target site. And we can use an activator here so that we can form a CRISPR activation, that is CRISPR A, so that it can help. Now we can also use the inhibitor to inactivate this portion, and we can also use the green fluorescent protein so that we can see the fluorescence of the and we can visualize the target portion where it is cleaving the target site. Now looking into the structure of the single guide RNA, so we have here the different portions. One is called as this gray portion, and the another is the yellow box portion. So this gray box here, it indicates the extra repeat anti-repeat region. So we have here the CRISPR RNA repeat region, and then we have the tracer RNA anti-repeat region in this whole single guide RNA scaffold. And this is usually truncated in designing single guide RNAs for genomic engineering. Now this yellow box, it denotes the 3' tail of single guide RNA, and this is not essential for Cas9 function, and it was omitted in the single guide RNA bound structure. So here we have also the region that is called as the seed region, which is approximately 10 to 12 nucleotides. So this is the seed region, and we have also the 20 nucleotides guide region, that is spacer region. And we have different loops, stem loops. One is the stem loop 1, another is the stem loop 2, and then we have the stem loop 3. So these are the different stem loops, and then we have also the linker portion which links the two loops, that is stem loop 1 and the stem loop 2. So the mismatches in this seed region, they can severely impair or abrogate target DNA binding and cleavage, and close homology in the seed region can often lead to off-target binding events, even with many mismatches elsewhere. And now we can see Cas9 makes extensive interactions with the single guide RNA. So the Cas9 enzyme, it makes the extensive interactions with the single guide RNA. Specifically, it makes many direct contacts with the repeat, anti-repeat, duplex, and stem loop 1, and as well as the linker region that is between the stem loop 1 and stem loop 2 through the helices 1, that is the arginine rich bridge helix, and the C-terminal domain. So this is the C-terminal domain here, and the Cas9 makes much less extensive contacts with the stem loop 2 of the single guide RNA. So this stem loop 2, it will have the less extensive contacts with the Cas9 enzyme, and primarily through its RUEC and the C-terminal domains. It uses the C-terminal domain and the RUEC domains so that it can interact with this repeat and anti-repeat duplex.